who's Dr. Ginny Espina, and she's an associate professor and she's director of the CAP-CLIA certified lab that you heard about, which she built from scratch when we came here to set up our center at Mason. She has a master's degree from Hopkins and she has her PhD from Mason. Take it away, Jenny.
So my topic is preventing breast cancer before it starts. So the first question I'd like to ask everybody to think about is, why are breasts important? Next. I'm sure everybody has their own opinion, but from a biological standpoint, breasts provide nutritive and immune functions. Breast cells secrete proteins, water, minerals, and antibacterial enzymes. One theory postulates that breasts evolved from sweat glands into these complex tissues composed of a convoluted system of milk ducts with fat, lymph vessels, immune cells, and connective tissue. And at puberty, as shown in this mouse model, mammary gland invasion commences in preparation for pregnancy and lactation, followed by involution after weaning. So invasion is a perfectly normal process in breast physiology. Next. However, when this normal process of invasion and involution goes awry, breast epithelial cells begin to accumulate within the milk duct at pathologically defined stages, which are ductal hyperplasia, which is just more cells than normal, atypical ductal hyperplasia, in which there are more cells, but the cells also start to look differently. They take on abnormal characteristics. The third stage is ductal carcinoma in situ, in which the cells have hallmarks of cancer. The fourth stage is DCIS with microinvasion, in which a few cells are starting to break out of the milk duct. And the final stage is an invasive ductal cancer. A breast duct that has, at the DCIS stage, can be 4 times the diameter of a normal breast duct. And this brings up 2 questions. The first one of which is, at what stage of this accumulation of cells within the milk duct do the cells possess the ability to invade? And if we figure that out, can we prevent breast cancer from becoming invasive? Next. To answer this question, we had to start with the simple process of, can we even grow human DCIS cells in the lab? This had never been done before. Next. We established a workflow between the patient, surgery, pathology, and the research lab. The breast lumpectomy specimen shown here, it has red and yellow and blue coloration to it. That's just ink that was applied by the pathologist to provide an anatomical orientation to the specimen related back to the orientation in the patient. And within this workflow, we were able to confirm the diagnosis of DCIS to assure a sense of quality control, but we were also able to cut the breast tissue into tiny pieces and actually grow them as organoids in the lab. Next. Next. So these organoids spontaneously formed spheroids and duct-like structures shown by the blue arrows from many different patients. And because of my background in the clinical lab, I knew we had to do this over and over again to show reproducibility. And this was a reproducible finding in many different patients. Next. To definitively demonstrate that these spheroids were tumor cells, we used a NOD SCID, a non-obese diabetic severe combined immunodeficiency mouse model. To confirm that the mouse lesion that we saw growing from the spheroids was actually a tumor and not just inflammation, we stained a tissue section with an antibody to human-specific cells called EpCAM, which is epithelial cell adhesion and activating molecule. EpCAM is shown here in green, proving to us that this was an actual tumor and not just an inflammatory reaction in the mouse. Next. The next question is, after we grew these cells, what molecular defects allow DCIS cells to thrive in the milk duct? Next. We know that genetic alterations are a hallmark of cancer cells. So using the Illumina 300,000 cytosnip bead chip, we were able to do molecular karyotyping or genotyping on the breast cancer tissue, the spheroids, the 3D structures that grew out. And what this chart shows you is each line represents a chromosome, and the color of the line represents the genetic alterations that we found. Red is a homozygous deletion, meaning you're missing 2 copies of that gene. Orange is a hemizygous deletion, you're missing 1 copy. Blue is a amplification or duplication of a gene. And green is a copy neutral loss of heterozygosity. Green is considered a neutral alteration and doesn't affect the carcinoma or the genetic status of the cells. Next. And as Dr. Leota and Dr. Petricoin emphasized, proteins are important. Why are they important? Well, as my daughter told me when she was 8 years old and she saw me working at home and asked what I was doing, she said, I get it. Proteins are the verbs of the cell. They do all the work. And so studying the proteins by reverse phase protein array technology allows us to quantify the amount of a protein and its post-translational modifications in a given tissue, a given sample for a given patient. Using this technology, our breakthrough discovery was that the DCIS spheroids are addicted to autophagy pathway for cell survival. Autophagy was not active in the epithelial cells or the cuboidal cells that also grew in our organoids. Next. So what is autophagy? Autophagy means self-eating. It's a perfectly normal digestive process in the cell. And autophagy occurs when a cell is under stress. It occurs when there's a lack of nutrients. Many of you may be upregulating autophagy right now if you're hungry. Autophagy allows cells to essentially eat snacks to make energy in the crowded milk duct. Next. Can we use a drug to prevent invasion or decrease the growth of the cells? All of our findings in the lab are only useful if we can actually translate this information to a patient. And is there a drug, more importantly, that's safe and effective for patients with DCIS? Next. So during autophagy, this cartoon shows the milieu of what is happening in the cell. A double membrane vesicle called an autophagosome forms, and it gobbles up proteins and extra organelles, and it shuttles them to the lysosome where they're digested, and the digestion then creates building blocks, amino acids, proteins that can be used further for cell survival. What we found, though, was that the cells, the DCIS cells, are addicted to this pathway. It's not cyclical in DCIS cells. It's always turned on, so these cells are always finding a way to survive in the hypoxic, nutrient-deprived milk duct. Next. So what can we use as a treatment or as a way to inhibit autophagy? Well, chloroquine, a drug that's been in the news because of COVID, is actually very effective in inhibiting autophagy. Chloroquine has been used in millions of people worldwide to treat malaria, lupus, and rheumatoid arthritis. Chloroquine has a very well-known toxicity and safety profile. And importantly, it's absorbed by the cell and transported into the lysosome, where it increases the pH, interfering with digestive enzymes, very similar to taking an antacid if you have an upset stomach. Next. And to ensure that we weren't inducing any further genetic abnormalities with the treatment, we treated the DCIS organoids growing in the lab with chloroquine, and repeated our molecular karyotyping, the surviving cells post-chloroquine treatment had fewer deleterious genetic mutations compared to the untreated spheroids. Next. To translate these findings to the clinic, we established a phase 1/2 clinical trial with Dr. Kirsten Edmiston at Inova Fairfax Hospital. The participants received either the standard malarial prophylaxis dose of chloroquine, which is 500 milligrams, or half that dose once a week for 4 weeks prior to their standard of care surgery. We analyzed by immunohistochemistry, tissue organoids, cytogenetics, and reverse phase protein arrays, the tissue before and after treatment. And this trial design allows us to assess the efficacy of treatment in a very short time period, only 4 weeks, rather than waiting for years for a typical preventive or treatment trial. And the unique aspect is each patient serves as their own control because we measure the tissue before and after treatment. The purposes of this trial were threefold. One was to test the hypothesis that by inhibiting autophagy in DCIS, we will reduce the capacity for DCIS to invade. The second was to test the safety and efficacy of chloroquine in DCIS patients because this hadn't been done before. And third was to evaluate whether chloroquine would reduce the DCIS lesion size as judged by MRI and histology, and if it would suppress the proliferation of the cells. Next. We only had 12 participants enrolled in the PINK trial due to a series of difficulties in recruiting patients. However, even though this is a small and underpowered study, 75% of the patients showed a reduction in the size of their DCIS lesion and a reduction in the proliferation index. The remaining 25% had a stable lesion size, but also had a reduction in the proliferation index. And so this PINK trial has provided data and methodology for additional clinical research with other compounds, either alone or in combination with chloroquine. Next. And I'd like to leave you with a thought. Imagine a future in which we could prevent the emergence of ductal carcinoma in situ by administering chloroquine or maybe another compound periodically to inhibit emerging cancer cells.